Sudden oak death is an invasive exotic forest disease that we have here in Oregon. It's caused by the pathogen Phytophthora ramorum, and our Oregon Tanoaks are most susceptible to this disease. It's found in southwestern Oregon here, and mostly along our coastal areas in Curry, and it stretches up into Coos, Josephine, and Douglas counties. Although tan oak is not a timber tree per se, it is highly important ecologically in our Oregon forest, providing food in terms of acorns and then also shade for our streams. Tan oak is a culturally important tree as well. Our native tribes here in Oregon have used many parts of tan oaks throughout their history. They use acorns as a food source, as well as their bark for tanning leathers and such.
Sudden oak death is a foliar pathogen. So it's a disease of the upper parts of tan oak trees. And basically what it does is it infects the leaves and the branches and produces spores, and those spores leak down the branches into the main stem, and they eventually kill the tan oak tree. Sudden oak death was first noticed in California, in the Bay Area. It was affecting coast live oak. And so these trees were dying, and no one really knew what was going on or what the problem was, and it took a few years to figure it out. And that started in around 1995. And then by 2001, they figured out what this pathogen was, and that's when they first identified it in Oregon. U.S. Forest Service and Oregon Department of Forestry conducted aerial surveys. They flew over most of the forests of southwestern Oregon, and they noticed some red, dead tan oak trees. Then a ground crew went in and they sampled those trees, and the samples were brought back to OSU, where the cause of the mortality was identified, and that was Phytophthora ramorum, which is the cause of sudden oak death.
So sudden oak death causes death of tan oaks once the trees become infected, and so we do have areas in Curry County where we have 100% mortality of tan oaks.
There's places in California where they say 99 or even 100% of the trees have been killed. Under suitable conditions, most of the tan oak could die if it's under suitable conditions for the pathogen.
So it has a really wide host range. So when I talk about host range, what I mean is the number of species that this pathogen is able to infect. And there are approximately 130 different species that Phytophthora ramorum, the cause of sudden oak death, can infect. In Oregon, the main species of concern is tan oak, but it also has been known to infect Douglas fir, it'll infect grand fir for tree species, and then it'll also affect a wide range of understory plant species. The advantage Oregon had is that we identified it early on in the outbreak. There were only a couple dead trees that were found on the landscape, and so that presented the opportunity to potentially actively manage this disease. And so a group of collaborators, Oregon Department of Agriculture, Oregon Department of Forestry, U.S. Forest Service, and OSU got together to develop a plan to help mitigate the impacts of this disease. The early stages of the sudden oak death program were eradication. Once the disease began to spread and occupy a larger area, we moved towards more of a slow the spread approach. So the idea here is that we would still actively manage the disease, try and reduce the rate that it spreads and kills more tan oak trees. A second phase of this approach was then to start thinking about developing resistant tan oak. And so that's sort of the phase that we're in right now, where we are working towards identifying resistance in the native tan oak population.
So we started a screening program, but it was vital with three groups. Oregon Department of Forestry, because they did the seed collections, they knew the field sites very well. BLM stepped up with some funding for the first 2 years, so otherwise the program would not have happened. Oregon State University was able to do a short-term screening, so they took that role on with some funding we provided them from BLM and later from the Forest Service. And then Dorena took on growing the seedlings, planning the whole initiative, and going forth. And so at this stage, we're still at a very early stage in screening. And so what we want to find out, is there resistance? What is the frequency of resistance? What are the types of resistance that are out there, so that hopefully we can put together an action plan.
Working with Oregon Department of Forestry, they were critical to get the ball going. They kind of They were able to help find land for our field disease testing. They're able to help with acorn collection, and they do a lot of work with eradication and understanding of sudden oak death on specifically tan oak.
The goal of a tree resistance breeding program is to use traditional plant breeding techniques in order to breed a tree that is resistant to a specific disease. So in this case, we're trying to find and locate tan oaks within southwestern Oregon that show some resistance to sudden oak death to be able to then put them back on the landscape.
It may turn out like with chestnut back east, and some people think it will be like that, where there's very, 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 very little resistance. There's different levels of resistance. We could find let's say resistance occurs on a 0 to 100 scale in terms of effectiveness. Well, if we find something with a 3 or 4, is that meaningful? You know, and it might be okay, hey, technically we have resistance, but maybe when we plant it out there, it dies a week later or a year later and stuff like that too. So we want to find resistance that's also meaningful and at a high enough level so that it will be durable and stable over different environments.
The project here in Oregon really began in earnest in 2016 with our first collection of tan oak acorns.
Alan Kanaski in ODF, who actually made the field collections. Alan knew some good sites for that, and he was able to do that.
So the acorns that we've collected so far have both been within the sudden oak death quarantine area, within infested sites, and both outside the sudden oak death quarantine area to get more of a range-wide collections of tan oaks. They've been small collections the last 3 years, but we're working with our collaborators on creating more of a range-wide collection of tan oak acorns and then focusing on survivor trees within the generally infested area just outside of the city of Brookings to see what resistance we can find. So the acorns are first assessed to see if they're viable or not, and then all of the acorns are given to Dorena Genetic Resource Center to grow up into seedlings.
So roughly over the past 3 years, I believe over 15,000 tan oak acorns have come to Dorena Genetic Resource Center to be grown. Over 7,000, I believe, more than 7,000 tan oaks have been actually grown at Dorena Genetic Resource Center. These are just a few of the tan oaks that have been grown here. If they were collected in an area that has sudden oak death, they have to go through an extreme quarantine. The seeds have to be bleached. It's a large amount of work. If they didn't, they come in. We give them each labels for which parent tree they're collected. So each parent tree needs to be kept separate. Once they get brought in, tan oak acorns can't be stored. They can't be put into a freezer. They need to be grown right away. So that's a really difficult thing with tan oak, unlike other tree species. So they're 1-year-old seedlings. And you can see that they have like unique identifiers on them for each tree. They've come from all different locations, and we made sure that information, that parent tree, we can track back to. And so if we found this seedling to have resistance, we can get back to that parent tree and collect more acorns.
To determine if seedlings are resistant or susceptible to this disease, we basically grow the pathogen which causes the disease on this petri dish, and we take the seedlings that were grown by Dorena, the Genetic Resource Center of the U.S. Forest Service, and we remove a single leaf, and then we place a piece of the pathogen that causes the disease on that wound, and then we wrap that in this wax tape. A week later, we come back and we measure how big of an area was killed by the pathogen. Seedlings with really big lesions would be considered really susceptible. Seedlings with smaller lesions would be considered to have some degree of resistance. And if no lesion developed at all, that seedling would be considered immune or totally resistant to the disease. We've already tested about 10,000 seedlings, and we've definitely seen variation in how resistant or susceptible these seedlings are, but we haven't found something that's totally immune yet. And so I'd imagine we're gonna have to test thousands more before we know. If we think about other invasive pathogens, that's sort of in line with what we have to do to identify resistance.
So we have 2 genetic conservation plantings outside of the disease zone. Those are just to conserve the genetics, being able to get back to those trees and utilize them either for DNA testing, trees that aren't impacted or don't have the pathogen on them. And we can use them for other research such as grafting or cloning, being able to take cuttings from them just as a resource. The other planting was planted in a disease zone outside of Brookings, Oregon, and it was to actually screen for disease resistance. We planted roughly 1,000 Tanoak seedlings in an area that was highly impacted by sudden oak death. And we've gone now 3 inspections, and we rate each seedling, all of the 1,000 seedlings for disease resistance. We wanted to find something that was at the cusp of where the pathogen was coming in, and so we still had living trees that could provide the spores that would inoculate what would cause disease on these plants. And so Sarah Navarro was critical to finding this field location. The planting, actually, it was a big group effort of Oregon Department of Forestry, Dorena Genetic Resource Center, so here, Forest Service with plant health protection, and then Oregon State University. So all of us together were out there digging holes, planting the plants. It was a large amount of fun, but also effort.
What we hope to learn from that is to see how these trees respond to inoculum in the field in Curry County, so natural infections, how they respond under lab conditions, because we're able to test more trees at a faster rate under lab conditions. And then the trees here at Schroeder, we just want to see, will they grow in the Willamette Valley? Can we potentially use this as a resource for future resistant stock to preserve the genes? So this is a 3rd program in Oregon that is breeding for resistance against a potentially devastating disease. The first being white pines. That program has been going on for quite some time, and then Porterford cedar, looking at breeding against Porterford cedar root disease, and now adding Tanoak into that.
In plant breeding, you say you need to grow thousands to find possibly one, the best. And so we need to go through numbers, and until you continue to screen more, you're never going to know. There might be 99.9% of them that die, but there is still that 0.1% that will live. And so the more we can screen, the more numbers we can get out there in field plantings and in OSU working in artificial inoculation. The more we can go through, the more likelihood we'll find that one living tree.